Ancient chemistry: why living things use TP as the universal energy currency. An early step in metabolic evolution set the stage for emergence of TP as the universal energy carrier. A simple two-carbon compound may have been a crucial player in the evolution of metabolism before the advent of cells. This is according to a new study by Nick Lane and colleagues of University College London, UK that was published in the open access journal PLOS Biology on October 4. The discovery may provide key insight into the earliest stages of probiotic biochemistry. In addition, the finding suggests how TP came to be the universal energy carrier of all cellular life today. Adenosine triphosphate is an organic compound that provides energy to drive many processes in living cells, such as nerve impulse propagation, muscle contraction, condensate dissolution, and chemical synthesis. ATP is found in all known forms of life and is often referred to as the molecular unit of currency of intracellular energy transfer. TP is used by all cells as an energy intermediate. During cellular respiration, energy is captured when a phosphate is added to ADP to generate TP. Cleavage of that phosphate releases energy to power most types of cellular function. However, building TP's complex chemical structure from scratch is energy-intensive and requires 6 separate TP-driven steps. While convincing models do allow for prebiotic formation of the TP skeleton without energy from already formed TP, they also indicate that TP was likely quite scarce. This means that some other compound may have played a central role in the conversion of ADP to TP at this stage of evolution. The most likely candidate, Lane and colleagues believed, was the two-carbon compound acetyl phosphate, which functions today in both bacteria and archaea as a metabolic intermediate. ACP has been shown to phosphorylate ADP to TP in water in the presence of iron ions, but a host of questions remained after that demonstration, including whether other small molecules might work as well. Whether ACP is specific for ADP or instead could function just as well with diphosphates of other nucleosides, and whether iron is unique in its ability to catalyze ADP phosphorylation in water. The authors explored all these questions in their new study. Drawing on data and hypotheses about the chemical conditions of the Earth before life arose, they tested the ability of other ions and minerals to catalyze TP formation in water. None were nearly as effective as iron. Next, they tested a panel of other small organic molecules for their ability to phosphorylate ADP. None were as effective as ACP, and only one other had any significant activity at all. Finally, they showed that none of the other nucleoside diphosphates accepted a phosphate from ACP. Combining these results with molecular dynamic modeling, the authors propose a mechanistic explanation for the specificity of the ADP-ACP iron reaction, hypothesizing that the small diameter and high charge density of the iron ion, combined with the conformation of the intermediate formed when the three come together, provide a just right geometry that allows ACP's phosphate to switch partners, forming TP. Our results suggest that ACP is the most plausible precursor to TP as a biological phosphorylator, Lane says, and that the emergence of TP as the universal energy currency of the cell was not the result of a frozen accident, but arose from the unique interactions of ADP and ACP. Over time, with the emergence of suitable catalysts, TP could eventually displace ACP as a ubiquitous phosphate donor and promote the polymerization of amino acids and nucleotides to form RNA, DNA, and proteins. Lead author Silvana Pena adds, TP is so central to metabolism that I thought it might be possible to form it from ADP under prebiotic conditions. But I also thought that several phosphorylating agents and metal ion catalysts would work, especially those conserved in life. It was very surprising to discover the reaction is so selective in the metal ion, phosphate donor, and substrate with molecules that life still uses. The fact that this happens best in water under mild, life-compatible conditions is really quite significant for the origin of life. Thanks for watching. Stay tuned with us to explore more science news.